Hi guys, and welcome to chapter 22. This chapter is also divided into three parts. It's all on the digestive system. Uh, pa- Kind of throughout the parts is a lot of anatomy reviewing the digestive system. So what I'm going to do is pick out some of the physiology concepts that I want you guys to focus on. You guys are responsible, like always, for all of the information within these three parts of the chapter, but I'm going to focus on some of the physiological processes of digestion. So with that, we're going to start in part B, and we're going to start in the stomach. Because again, up until this point, it's a lot of anatomy review. There's some function involved, but I'm going to focus and we're going to start on the microscopic anatomy of the stomach. So before this, I would encourage you to maybe pause and review the slides up until this point, as well as part A, because it kind of gives you a good overview of all the anatomy of the GI tract up until this point. So as you remember, your stomach contains four layers or tunics. However, the muscularis and mucosa layer of the stomach are modified in comparison to the other GI tract layers. The muscularis externa has circular and longitudinal smooth muscle layers, as well as an extra third layer, which is what we call the oblique layer. Together, the smooth muscles of your stomach allow the stomach to not only churn, mix, and move the chyme, which is the word for the food. So once the food reaches your stomach, we call it chyme. This extra layer allows the stomach to pummel it, which increases the physical breakdown and kind of ramming it into the small intestine. The mucosa layer is also modified. It consists of simple columnar epithelium entirely composed of mucus cells, which will secrete a two-layer coat of alkaline mucus. And this surface layer will trap bicarbonate-rich fluid that's beneath it. The microscopic anatomy of the stomach, you'll have gastric pits, which lead into gastric glands. And gastric glands are what will produce the gastric juice to help with digestion of the stomach contents. So here's a look at the layers of the stomach wall. And you can see here the gastric pits are lined with gastric glands and the layers below the stomach. I do want you guys to know the different types of gland cells. So the glands in the fundus region of the stomach and the body produce the most gastric juices. And the glands include secretory cells, which I want you guys to know, and I want you to know what they secrete. The mucous neck cells, the parietal cells, the chief cells, and then the enteroendocrine cells. So here is a look at where these cells are located within the gastric pits and the gastric glands, the mucous neck cells, parietal cells, chief cells, and then the enlarged view of the gastric pits and gastric glands. So what do all of these cells do in these glands? The mucous neck cells secrete a thin acidic mucus of unknown function. The parietal cells are very important because they secrete hydrochloric acid. And one thing that you should know is that the stomach is a very acidic environment because of your parietal cells secreting this hydrochloric acid. Hydrochloric acid is important because it denatures or breaks down protein. It also activates pepsin, which we'll talk about. It breaks down plant cells' walls. So any sort of vegetable or plants or lettuce that you eat, the hydrochloric acid will help break that down. HCl also kills many bacteria, which is a body defense mechanism. Intrinsic factors also released by your parietal cells, and glycoprotein required for the absorption of vitamin B12 is in the small intestine. So intrinsic factor is just a glycoprotein that helps to absorb vitamin B12 in the small intestine. Chief cells then secrete pepsinogen. This is an inactive enzyme which will be activated to pepsin by HCl and by pepsin itself. And chief cells also secrete lipases, which digest about 15% of your lipids. Enteroendocrine cells then secrete chemical messengers into the lamina propria, and they act as paracrines. So they secrete serotonin and histamine, and also they secrete hormones, which secrete somatostatin and gastrin. So this is just a review of the different cells of the gastric glands and what they secrete and why it's important. So the parietal cells secrete HCl, an intrinsic factor. Chief cells secrete pepsinogen, which will be activated by HCl that turns pepsinogen into pepsin. And then the enteroendocrine cells secrete hormones and other paracrines. The mucosal barrier is a barrier that helps to protect against the harsh digestive conditions that are required by the stomach to be protected. The mucosal barrier protects the stomach, is created by 3 different factors: the bicarbonate-rich mucus, the tight junctions, and then these damaged epithelial cells are quickly replaced. Gastritis then is an inflammation caused by anything that breaches the stomach's mucosal barrier. It can cause peptic or gastric ulcers, which are erosions in the stomach wall. And if erosions will perforate the wall, it can lead to peritonitis and a hemorrhage. Most ulcers are actually caused by a bacteria, but they can also be caused by NSAIDs, such as aspirin. So this is a look at a gastric ulcer. It looks shiny and red. You can see the triangular dark hole that pierces through the stomach wall. It's just as painful as it looks. Here's a look at a bacteria attaching to the mucosa layer in the stomach, which could eventually cause an ulcer as well. So then the digestive processes in the stomach, processes carried out by the stomach. It helps to break down your food. Your stomach serves as a holding area for the food. It delivers chyme eventually into the small intestine. It denatures proteins by hydrochloric acid. And then pepsin carries out the enzymatic digestion of proteins. The lipid-soluble alcohol and aspirin are absorbed into the blood in the stomach, and the only stomach function essential to life is the secretion of intrinsic factor for vitamin B12 absorption in the small intestine. What is vitamin B12 important for? It's needed for red blood cells to mature, and if we don't have intrinsic factor, it can cause pernicious anemia, which can be treated with B12 injections. So these are some of the hormones and the paracrines that act in digestion. We'll go over some of these a little more when we get to part C of this PowerPoint of digestion as well. Here are the peristaltic waves in the stomach. So the smooth muscle layers in the stomach help to propel the chyme. They grind up the chyme. And then the retropropulsion just means it kind of moves it back and forth to mix it. Regulation of gastric emptying. The duodenum can prevent overfilling of the stomach by controlling how much chyme enters it. The duodenal receptors respond to stretch and chemical signals. The enterogastric reflex and enterogastrones inhibit gastric secretions and duodenal filling. And the stomach empties in about 4 hours. But if there's more fatty chyme entering the duodenum, it can increase the emptying of the stomach into 6 hours or more. So this is a look at the neural and hormonal factors that will inhibit gastric emptying. Vomiting, also called emesis, is caused by extreme stretching of the stomach. It can also be caused by any sort of irritant, like a bacteria, excessive alcohol, spicy food, certain drugs. Chemicals and sensory impulses can stimulate the emetic or vomiting center in the brain, and excessive vomiting can lead to dehydration or electrolyte and acid-base imbalances, such as alkalosis, which is a more basic imbalance. The liver, gallbladder, and pancreas then are accessory organs to the GI tract. They're associated with the small intestine, and I want you guys to have a really good understanding of these 3 organs. The liver's main digestive function is to produce bile, and bile helps to break down fat. The main function of the gallbladder is to just store the bile, and the pancreas supplies most of the enzymes needed to digest chyme, as well as supplying bicarbonate to neutralize the stomach acid. Here's the gross anatomy of the liver, which I'll have you guys review on your own. There's a look at the liver with its lobes and its ligaments. More gross anatomy of the liver. It has bile ducts. The common hepatic duct leaves the liver. The cystic duct connects to the gallbladder. And then the bile duct is formed by the union of the common hepatic and the cystic ducts. So understanding the duct system entering and exiting the liver is important, as well as the ducts entering the gallbladder. The liver, a little bit microscopic anatomy, it's formed into liver lobules, which are hexagonal structural and functional units. These lobules are composed of plates of hepatocytes, which are liver cells, and you have a central vein located in the longitudinal axis. The portal triad refers to each corner of the lobule, which contains a branch of your hepatic artery, the hepatic portal vein, which brings nutrient-rich blood from the intestine, and then the bile duct, which receives bile from the bile canaliculi. So here's a look at the lobule, and each corner of the lobule will contain these portal triads. There's a central vein, connective tissue surrounding each. Liver sinusoids are the leaky capillaries located between the hepatic plates. And then you have macrophages within these sinusoids, which remove debris and old red blood cells. So this is a good picture to kind of study. So the portal veins are always kind of carrying nutrient-rich blood from the intestines. You can see the arteries in red bringing oxygen, and then the bile ducts, which are receiving bile from the canaliculi, which are producing the bile. And then the bile ducts will eventually take the bile to the gallbladder to store. The hepatocytes have increased rough and smooth Golgi apparatus, peroxisomes, mitochondria. The big functions of your hepatocytes are to produce bile, process bloodborne nutrients, and also to store glucose as glycogen, store fat-soluble vitamins, and also perform detoxification, such as converting ammonia to urea. Bile is the composition and enterohepatic circulation. It's a yellow-green substance. It contains bile salts and bilirubin. Bile salts are what will function to emulsify fat and absorb fat. Bilirubin is formed from heme. It's a bacteria that helps to break down in the intestine. And then you have cholesterol, triglycerides, phospholipids, and electrolytes. Bile is important in the enterohepatic circulation. Bile salts are reabsorbed into the blood, they return to the liver, and then they're re-secreted in newly formed bile. So we kind of get this recycling mechanism of bile. About 95% of secreted bile salts are recycled, so only 5% is newly synthesized each time. And this is a look at the enterohepatic circulation. So bile salts, number one, are secreted into the duodenum. As bile salts travel through the small intestine, they allow lipid digestion and absorption to occur. And now about 95% are reabsorbed in the ileum, the last part of the small intestine. And reabsorbed bile salts are then traveled back to the liver to be reused to create more bile salts. Some homeostatic imbalances of the liver, hepatitis is usually by a viral infection, drug toxicity, or wild mushroom poisoning. So that's an inflammation of the hepatocytes or the liver. Cirrhosis is a progressive chronic inflammation from chronic hepatitis or alcoholism. The liver can become fatty and fibrosis, which can lead to what we call portal hypertension, so kind of high blood pressure within the portal hepatic system. Liver transplants can be successful, but they're very scarce. Liver can regenerate, so it's one of the few organs that can regenerate to its full size in about 6 to 12 months after 80% is removed. The gallbladder then is a thin-walled muscular sac. It functions to store and concentrate bile. It contains many honeycomb folds that allow to expand as it fills. Gallstones, are caused by too much cholesterol or too few bile salts. Gallstones can be incredibly painful because they can obstruct the flow of bile from the gallbladder. It can be incredibly painful when the gallbladder contracts against the sharp crystals. Obstructive jaundice is what we call when there is a blockage that can cause bile salts and pigments to build up in the blood, resulting in a jaundiced or yellow skin. Jaundice can also be caused by liver failure. So for a gallstone treatment, you can take crystal dissolving drugs. You can try to vibrate through an ultrasound to get rid of the gallstones, laser vaporization, or surgery, which will just take out the gallstones and sometimes the whole gallbladder. The pancreas then is located mainly in the back retroperitoneal, the back abdominal cavity, deep to the greater curvature of the stomach. The exocrine functions of the pancreas are to produce pancreatic juice. So the acini, these are clusters of secretory cells that produce zymogen granules containing proenzymes. And then the ducts will secrete all the pancreatic juice to the duodenum via the main pancreatic duct. And there's also a smaller duct that helps to produce water and bicarbonate. The endocrine functions of the pancreas are to secrete insulin and glucagon by pancreatic islet cells. So here's a look at the structure of the enzyme-producing tissues of the pancreas. You can see the acinar cell, the zymogen granules, the rough endoplasmic reticulum. This is one acinus, and it will secrete its pancreatic juice or fluids into the duct cell. This is an actual histological microscopic view of the enzyme-producing tissues of the pancreas, the acinar cells, and then the pancreatic ducts. So the pancreas, the important function of it is to produce pancreatic juice. This pancreatic juice is alkaline, so it has a pH of 8, a basic pH, and this helps to neutralize the acidic chyme coming into the small intestine from the stomach. It also includes electrolytes, digestive enzymes, proteases, which will break down proteins, amylase, which will break down carbohydrates, lipases, breaking down lipids, and nucleases, which are breaking down nucleic acids. So here's a look at the pancreas with the pancreatic duct entering the duodenum. And you can see here the things entering the duodenum are trypsinogen, cryptotrypsinogen, and procarboxypeptidase, which are all inactive and then will be activated by substances from the epithelial cells of the small intestine into its activated form. The bile duct and pancreatic duct unite in the wall of the duodenum to form this ampulla. And the ampulla opens up into what we call a papilla. There's a sphincter that controls the entry of bile and the pancreatic juice into the duodenum. And the accessory pancreatic duct is just a smaller duct that also empties into the duodenum. So here's a look at the duct system entering into the major duodenal papilla. We have sphincters that control the secretions of bile entering and the pancreatic juices entering. Here you can see the common hepatic duct joining up with the cystic duct from the gallbladder to form the bile duct. So we have bile and the pancreatic juices both entering through this major duodenal papilla. You can see a smaller entry point from an accessory pancreatic duct, but most of the bile and digestive enzymes that come from the pancreas are entering through this major duodenal papilla, and that is controlled by sphincters. Regulation of bile and pancreatic secretions are both stimulated by neural and hormonal controls. These are the hormonal controls, cholecystokinin, CCK, and secretin, which are again controlling the digestive juices entering into the duodenum. Bile secretion will be increased when the enterohepatic circulation returns large amounts of bile salts. Secretin will also stimulate the gallbladder to release bile, and then the hepatopancreatic sphincter will be closed unless digestion is active. So here's a look at the mechanisms promoting secretion and release of bile and pancreatic juices. CCK and secretin are secreted by the duodenal enteroendocrine cells. Pancreatic secretion, we have CCK, secretin, vagus nerve, all controlling the secretions of these digestive juices. Bile secretion by the liver, bile salts return from the enterohepatic circulation. Secretin is a minor stimulus, gallbladder contractions, and then the hepatopancreatic sphincter relaxation. That has to do with the sphincter, which will relax or open to allow all of these juices and bile to enter the duodenum. So this is the part B, and then we're going to move right into part C. So we finished with kind of part B, and now we're going to continue with part C. Part C up until this point includes a lot of anatomy review. So we're going to pick up with part 3, where we'll look into the physiology of digestion and absorption of all the nutrients that your body takes in. I'm just going to lower this down so I can make it a little bigger here. So mechanisms of digestion and absorption. So the word digestion, that has to do with breaking down ingested food into its chemical building blocks, and only 3 molecules are small enough to be absorbed across the wall of the small intestine. Digestion is the catabolic process that breaks macromolecules down into monomers small enough for the absorption. You have intrinsic and accessory gland enzymes, which are involved in digestion. The enzymes carry out hydrolysis, where water is added to break down chemical bonds. Then the absorption is the process of moving these small substances from the lumen of the GI tract or the gut into the body. You have tight junctions, which ensure that the molecules must pass through epithelial cells rather between them. Materials can enter the cell through the apical membrane, the lumen side, and exit through the basolateral membrane, the blood side. Lipid molecules can be absorbed passively through the membrane, but other polar molecules have to be absorbed by active transport, and most nutrients are absorbed before the chyme reaches the ileum of the small intestine. So here's a look, again, just showing the mechanisms of absorption. Here's a villi of the small intestine, kind of a finger outpocketing. We're going to zoom in on kind of the wall of a villus. The apical membrane includes the brush border microvilli, and this shows the route of absorption through the enterocyte, or one of the cells. Here's the basolateral membrane, and you can see here the route of absorption. We want to go through the cell into the blood capillary. So through the interstitial fluid, eventually into the blood capillary, into the vein that will take all the digested and absorbed materials into the liver for more processing. So the processing of nutrients then, first of all, we'll talk about carbohydrates. Only monosaccharides can be absorbed, and a monosaccharide is a single monomer of a carbohydrate, so it's a single sugar. Starch and disaccharides will be broken down to oligosaccharides and disaccharides, and this begins in the mouth with salivary amylase. They will be further broken down into lactose, maltose, and sucrose. And the final breakdown is into a monosaccharide. And monosaccharides, again, are just the single sugars. These are glucose, fructose, and galactose. So this shows the flow chart of digestion and absorption of foodstuffs. So you can see here where it starts in the mouth, where salivary amylase starts the breakdown of carbohydrates. Pancreatic amylase from the pancreas further breaks down kind of the several chained sugars. And eventually the goal in carbohydrate digestion is to break down these many chained sugars into their single sugar, whether it's galactose, glucose, or fructose, because the monomer, galactose, glucose, and fructose, are the only structures that can be absorbed into the bloodstream within the small intestine. So the steps of starch digestion in the intestine includes pancreatic amylase, which breaks down starch or glycogen that escaped the salivary amylase into an oligosaccharide and a disaccharide. The brush border enzymes in the small intestine will further break these into lactose, maltose, and sucrose, and then eventually into a monosaccharide, a single sugar, which is glucose, fructose, and galactose. Monosaccharides are then co-transported across the apical membrane of these absorptive epithelial cells, mostly by what we call secondary active transport with sodium. And then monosaccharides will exit across the basolateral membrane by facilitated diffusion. So I'll show a picture here. This shows the long-chained starch. Pancreatic amylase breaks down starch and glycogen into an oligosaccharide and a disaccharide. The brush border enzymes break oligosaccharides and disaccharides into monosaccharides. And again, the monosaccharides, which are these single kind of hexagonal shaped structures, those are what can be absorbed through the cell. So you'll only notice these single hexagon, single sugars being able to be taken up into the cell and then transported across the cell. So these monosaccharides, glucose, galactose, are co-transported across the apical membrane of the enterocyte. And this active transport uses sodium concentration gradient established by a sodium-potassium ATPase pump. So this is another part of the body where you need ATP. into the basolateral membrane shown here. Finally, then these monosaccharides exit that cell, and then they cross the basolateral membrane by facilitated diffusion and eventually enter the capillary via the intercellular clefts. So again, the goal from the food that you eat is you want to break it down into a monomer, whether that's glucose, and glucose eventually needs to be absorbed into the bloodstream, because then glucose will be taken to the liver for processing, whether it will be stored there, or glucose will be turned into ATP, which we'll learn about in chapter 23. People with a lactose intolerance have a deficient amount of lactose and cannot consume lactose. Any lactose eaten remains undigested and can create an osmotic gradient in the intestine. What that does is it will prevent water from being absorbed, resulting in diarrhea. It can also pull water from the interstitial space back into the intestinal lumen. So basically, people with lactose intolerant aren't able to absorb water, so they just kind of get rid of the water through diarrhea. Bacterial metabolism of undigested solutes can produce large amounts of gas, resulting in bloating, flatulence, and cramping of pain. And the treatment is to add lactase enzyme drops to milk or to take a lactase tablet perform consuming milk products to help digest the lactose. Then, how are proteins digested? So the source of protein is not only is dietary, but also includes digestive enzymes and proteins from the breakdown of mucosal cells. So proteins are broken into large polypeptides, small polypeptides, and small peptides, and finally into an amino acid monomer. The digestion begins in the stomach when pepsinogen is converted into pepsin at an extremely acidic pH levels, and it will become inactive in the high pH of the duodenum. So here's a look at protein digestion. It starts in the stomach, continues in the small intestine, and eventually proteins get broken into amino acids, which can be absorbed then in the small intestine. So the steps of protein digestion in the intestine, so your pancreatic proteases trypsin and chymotrypsin will cleave proteins into smaller peptides, while carboxypeptidase takes off one amino acid at a time from the end. You have a brush border enzyme, which helps to break these oligopeptides and dipeptides into their single amino acid component, because the single amino acid component are what can be digested through the small intestine. Amino acids then are co-transported, much like sugars are, across the apical membrane of the absorptive epithelial cell via a secondary active transport carrier. And then the amino acids can exit across the basolateral membrane via facilitated diffusion. So here's just a look again at how proteins are digested and absorbed in the small intestine. Pancreatic proteases are broken down into proteins and protein fragments into smaller pieces and some individual amino acids. The brush border enzymes break the protein fragments into the single amino acids. The amino acids are then co-transported across the apical membrane of the epithelial cell. This is an active transport process that uses the sodium concentration gradient established by sodium-potassium ATPase. And then the amino acids exit across the basolateral membrane via facilitated diffusion and enter the capillary via the intercellular cells. Then we'll talk about how lipids are digested. Lipids are digested in the intestine via what we call emulsification, which is where triglycerides and their breakdown products are insoluble in water. So they need a pre-treatment with bile salts that just help to break the large fat globules down into smaller ones. Digestion of lipids then uses pancreatic lipases, which will break down fat into a monoglyceride plus 2 free fatty acids. Then a missile is formed, not to be confused with a missile from a weapon, but a missile is formed, which means that products from the digestion become coated with bile salts and lectin, and then the lipid products can diffuse They leave the micelles and cross the epithelial membranes via diffusion. Chylomicron formation is when the lipid products are converted back into triglycerides and packaged into these lectin and lipoproteins, forming a chylomicron. The chylomicrons can then be transported and enclosed through the cell and eventually emptied into venous blood at the thoracic duct. Once in the blood, these chylomicrons are broken into free fatty acids and glycerol by lipoprotein lipase so that they can be used by cells. Short-chain fatty acids can diffuse directly into the blood. So let's take a look at the flowchart of digestion and absorption of the lipids then. Again, it starts in the mouth and then it continues with pancreatic lipases in the small intestine, and we'll go through the steps. So emulsification involves the bile salts in the duodenum breaking down fat globules into smaller fat droplets, increasing the surface area available to these lipase enzymes. The pancreatic lipases can then hydrolyze or break apart these triglycerides, yielding a monoglyceride and a free fatty acid. And then the micelle is formed, where these free fatty acids and monoglycerides assemble with the bile salts, forming micelles. Micelles can then ferry their contents to the enterocytes, and these can then be diffused. The fatty acids and monoglycerides can then be diffused from the micelles into the enterocytes. The chylomicrons are then formed, where the fatty acids and monoglycerides with other fatty substances and proteins form a chylomicron. And then the chylomicron can then be extruded from the enterocytes. And through exocytosis, they enter what we call the lacteal. And the lacteal is what will transport the fatty acids then to the intestine in the lymph. The nuclei of an ingested cell in the food can contain DNA and RNA, and pancreatic nucleases can hydrolyze or break down the nucleic acid to a nucleotide monomer. The brush border enzyme includes nucleodases and phosphatases, which will break down nucleotides down into free nitrogenous bases, pentose sugars, and phosphate ions. The breakdown products will be actively transported by special carriers in the epithelium of the villi. So this is a breakdown showing the DNA. Vitamin absorption then in the small intestine, fat-soluble vitamins are carried by micelles. Water-soluble vitamins are absorbed by diffusion or by passive or active transporters. Vitamin B12 is a large charged molecule which binds with intrinsic factor and is absorbed by endocytosis. And in the large intestine, vitamin K and B vitamins from bacterial metabolism will be absorbed. Electrolytes are also absorbed via different mechanisms that you can read through. Absorption of water, about 9 liters of water, most from GI tract secretions, enters the small intestine, and most of the water is absorbed in the small intestine by osmosis. Malabsorption of anything can be caused by anything that interferes with the delivery of bile or pancreatic juices. This can be caused from damaged intestinal mucosa, from a bacterial infection, sometimes an antibiotic. Gluten-sensitive enteropathy or celiac disease is also a common malabsorption disease. It's an immune reaction to gluten, where gluten causes the immune cell damage to an intestinal villi and brush border, and the treatment is to eliminate gluten from the diet. So again, this is chapter 22. There's a lot of information in chapter 2. I covered some of the physiological processes of digestion and absorption. You guys are responsible for all the information within the PowerPoints, but I wanted our video to kind of highlight some of the things that I want you guys to focus on. Thanks for listening, guys, and I'll see you for our next video.